All right, this video is for the MiniPCR CrimeLab. So this is your pre-lab video. What we're going to do is we're going to be using the MiniPCR thermocycler to uh, conduct some PCR and then use it to solve a crime. So to do a little background information, what we're going to do is we're going to review DNA structure and DNA amplification concepts and why we do that, and then of course solve the crime using PCR in the lab. Okay, this will be a 2-day lab. The first day we'll be doing the PCR, and then the second day we'll do the gel electrophoresis to interpret the results. Okay, so pretty much why is PCR important? Um, in order to really analyze our genetic sequences, we need to have many, many copies of the DNA. Well, as it turns out, PCR allows us to make numerous copies, or another word is to amplify our DNA sequence, and therefore we can analyze it better so it can be used in forensics and human evolution, molecular diagnostics, personalized medicine, such as like DNA chips, consumer genomics along the same lines, and then production of GMOs and use in food and agriculture. Okay? So just to kind of overall what PCR is, PCR stands for polymerase chain reaction. Right? So what we do is we end up taking your favorite gene, which is whatever it is, that little red sequence right there, and this is the sequence that we're wanting to analyze. We'll undergo, or we'll put it through PCR, and then with the PCR we make billions of copies of this DNA without ever having to put it into a cell. And then when we have those billions of copies of DNA, we can then use them for forensic analysis, sequencing, drug development. Okay, the list goes on and on. Okay, so the process that identifies and copies amplifies a specific piece of DNA is in a biological sample, right? So that's the whole purpose of this, is to take a biological sample and make billions of copies of it in 30 minutes, right? So just a little review of structure, right? DNA is a double helix composed of a sugar phosphate backbone, and those sugars are holding on to these nitrogenous bases, adenine, thymine, guanine, cytosine, right? Inside the rungs of this ladder, if we were to untwist it, we have G's and C's, A's and T's coming together, and what we do is we can identify a sequence of A's, T's, C's, and G's and develop methods to use PCR to amplify or make many, many copies of that particular sequence. Okay? Now, PCR works in 3 steps, right? A mini PCR machine, just like all of the PCR machines, they're called thermocyclers because they're going to cycle through 3 different temperatures because there are 3 different steps that are going on, right? The first step is called denaturation, in which the DNA is heated up to 94 degrees Celsius, and what happens is it's going to break the hydrogen bonds that are holding the nitrogen spaces together, right? And they're going to separate like so. And then in the second stage, the temperature cools down to a 50 to 60 degrees Celsius to allow the primers, right, these beginning pieces of DNA, to attach to the strand or the sequence that you're looking for. And we call that annealing, right? So the annealing is the process of attaching the primers. And then extension is when we have DNA polymerase come in and start using the free-floating nucleotides, these deoxyribonucleic triphosphates, to come in and start attaching to our open DNA strands. Right, our single-stranded DNA is now becoming double-stranded. Now that the primers have been attached, which allows DNA polymerase to come on and start making copies. Right, and that happens at 72 degrees Celsius. What's going to happen after it's done making this strand? It's going to heat back up to 94 degrees, and then it's going to split. Instead of 1 strand of DNA, it's going to split 2, and then 4, and then 8, so on and so forth. Okay? So, like I said, we just keep repeating the cycle. We'll repeat it 20 to 30 times, and then by the time it's all said and done, we'll have billions of copies of our DNA for either sequencing, or in this case, we're going to use it for forensics. And we'll use gel electrophoresis to identify the different strands of DNA. So, for example, we have this is a picture of a gel, right? These little bands are representing segments of DNA. The farther down you go, the smaller the strands are. The higher up you go, the bigger the strands are, right? there's a little segment right in here, right? You can see it right there, and you can see it over here. Those are DNA ladders, and we'll talk more about those when we do the gel electrophoresis portion of this lab. Okay, so today, or more likely tomorrow, we're going to do setup, and we're actually going to do the PCR, and then the next day we're going to do electrophoresis, and then we'll visualize what we, the fruits of our labor. To give you a little introduction into what MiniPCR looks like, it's about the size of a tissue box. Okay, I've shown it to you in class before, but we have two fans that are going to be using to these fans. This heating block are going to be used to warm up and cool down the system. Right, we have a microprocessor right here, which is going to allow us to send a program through an online software to allow us to tell us, okay, well, how long do I want it to be denaturing? How long do I want it to be annealing? How long do I want it to be how long do we be extending, right? So microprocessors used to control it, cooling fans and heating blocks just to heat up and cool things down, and then we have a heated lid to prevent condensation. Heating block, heated lid, guess what? They're hot, so be careful, okay? So we're going to use it to solve this mystery of Missy Baker is missing, and the police don't know what to do, so they're going to rely on you and your classmates to help them out, right? This is not an uncommon thing in our criminal justice system. We see DNA tests and the use of DNA technology to clear the names of people and also condemn them for whatever boo-boos they did, right? So here's our little problem. Missy Baker, gay pastry shop owner, goes missing. Police at a loss. You're more than welcome to read through this. But what you need to know is that Missy Baker has cystic fibrosis. Okay, now cystic fibrosis is it's a mutation in the CFTR gene, right? So a healthy person has a normal length CFTR gene, while somebody with cystic fibrosis, like Missy Baker, has a deletion in there. Now, of course, what happens is when this deletion, this CFTR gene is shorter, right? So obviously, if I have two strands of DNA that are shorter, or one strand of DNA that's shorter than the other one, then it's going to look different on electrophoresis gel. Okay, so keep that in mind. So here, for example, this is a little ladder right here that has mapped out 100 base pairs, 200 base pairs, up to 1,000 base pairs. Okay, take a wild guess as to which one of these individuals has cystic fibrosis. Okay, hopefully you pick the one on the right, because they have a shorter segment because it's only, what, 415, 410 base pairs, while the individual on the left has about 850 base pairs. So they do not have, this guy right here does not have the deletion, this person right here does. Okay? So what's going to go into this reaction, right? Okay, so we saw the big picture, but we need to know what's really going in there. Okay, so we need a template of DNA to be amplified, so we're going to, in this case, have some samples of some suspects, right? We have one suspect, and suspect A and suspect B. Suspect A and B both had some blonde hair, or what could be Missy Baker's hair, in their car. So what we're going to do is we're going to test that DNA sample to see if they have a CFTR deletion, right? And then we'll use PCR to make many, many copies of that gene, because if we don't, there's usually not enough DNA to really run a gel electrophoresis. The more DNA strands we have, the clearer the bands look, the more distinct our analysis can be, right? So we'll need a pair of DNA primers to attach to the open-stranded DNA during annealing, and then we'll need DNA polymerase for the extension portion where we're building the double-stranded molecule. And of course, in order to build the double-stranded molecule, we need the DNTPs, right? Deoxyribonucleotide triphosphates, right? Those are just another way of saying nucleotides, right? Those are going to be attaching to the parent strand, and then we'll be building over the original strand, and they'll keep building. We'll need a buffer to maintain pH, just to if we have something that's too acidic or too basic in the tube, that could interfere with the annealing process, not to mention the extension process, because obviously if it's too acidic, DNA Taq polymerase, which is this guy right here, may not be able to do its job because enzymes are specific like that, right? Very picky, those enzymes. And of course, we need a thermocycler to regulate the temperature, and this is our mini PCR machine right here. Okay. So what's going to go into the tubes? You're going to have 4 tubes, right? You're going to have tube A, tube B, somebody with a healthy gene and somebody with a deleted gene. So you have 2 controls. You have what it looks like when somebody has a normal gene and when somebody has a deletion, right? And this is our mutant, of course. And then we have the DNA found from suspect A's car, DNA found from suspect B's car, and hopefully one of them match up to the deleted mutant, because if that's the case, then we can identify where Missy Baker was, right? Now, these are the quantities that you need to put into your 4 PCR tubes, right? A, B, H, and D that you'll label. 5 microliters, 10 microliters, 10 microliters for a grand total of 25 microliters. Okay? These temperatures and the amount of times, they are given in the instructions, so don't worry about writing them down. We'll just pay attention when we get into class. Okay? This is what the software looks like, right? This is where you'll, so these numbers right here is where this is where you'll enter that data. Okay? Number of cycles. Okay? In this case down here, you actually, if you wanted to at another time, you could upload a predestined or predetermined program into it to determine or to have a set. But I mean, it's not really that big a deal just to enter these numbers. So as far as how we're going to monitor it, once we start it, we really don't do too much. But there's a couple things we should be thinking about. During denaturation, what's happening to the DNA molecules? During annealing, what's happening in the DNA molecules? During extension, what's happening? Okay. Now, why do we need to add the enzyme Taq polymerase? Right. Now, as it turns out, Taq DNA polymerase is somewhat unique. This is the little bit of homework I want you to do. What's so special about Taq polymerase? And of course, if you look at the question I have right above it, right, which that doesn't really help, what temperature is optimal for most enzymes? Obviously, temperature is going to have something to do with Taq polymerase. So tell me, what about Taq polymerase makes it so special, right? And then lastly, how many more molecules of DNA would we have after each PCR cycle and at the end of the entire PCR reaction? Okay, I do recommend that you go to, if you want to kind of see and do a dry run or a virtual run of this, you can always do go to Google and type in virtual PCR lab Utah genetics, okay? And then that should bring you to a virtual PCR lab, so you can actually do a PCR lab before you actually do one for real tomorrow, okay? And speaking of tomorrow, I'll see you tomorrow.